So today I will start with developmental biology, and specifically in the developmental biology, I'll try to cover spermatogenesis and a bit of oogenesis in today's class. These are easy topics, and you people will be able to do it very well. I have sent the notes already on WhatsApp. Please go read. Whenever you have questions, feel free to ask me. I'm sharing my board right now. Are you people able to see this board?
Yes,
Yes,
sir.
sir.
Yes,
Okay.
sir.
Okay. So let's start with spermatogenesis. So basically, it is the actually the formation of sperms or sperms. So properly saying, you can say that it is the process. It is a process, and in this process, what happens? Spermatozoa are produced, or sperms are produced. And from where are they produced? They are produced from, just hold on a minute. Somebody is trying to enter. So, from where are they produced? They are produced from primordial germ cells. And the process is mitosis and meiosis. So, the initial cells from which all these sperms are formed, they are called spermatogonia. They are called spermatogonia. And these spermatogonia, by the process of mitosis, they yield spermatocytes. These spermatogonia, they are throughout in the process of multiplication. They remain the process of multiplication. And this multiplication, this is through the process of the cell division of mitosis. Mitosis is also known as the equational division. Now, if you go through the process of spermatogenesis, just to make you aware, two chromosome sets. This is, say, this is the spermatogonium. This spermatogonium will give rise to it will duplicate. The univalents will duplicate. It will give rise to the primary spermatocytes. Clear? And this will keep on mitosis and multiply through mitosis. Keep on my lots of spermatogonium will take place. Now, through the process of meiosis 1, the spermatogonia will form secondary spermatocytes. They will form secondary spermatocytes. These are known as secondary spermatocytes. Now, these secondary spermatocytes again will undergo process of meiosis 2. It is similar to that of mitosis. It will form the spermatids. Aha. It will form spermatids. These are known as spermatids. Up to this stage, spermatids have been formed. Now, they will mature and form sperms or mature sperm cells or spermatozoa. These are mature sperm cells. And this is to show the whole thing together. Once more, spermatogonium to spermatocytes, spermatocytes to secondary spermatocytes, secondary spermatocytes to spermatids, spermatids to mature sperm cells. Clear? Yes, no, बोला था?
Yes, sir.
Yes, sir.
Yes, sir.
Yes, sir.
Okay,
Yes, sir.
thank you. Next is I'll explain some more things. Spermatogenesis is also known as it is the obviously it happens in males. It is the male version of gametogenesis. It is the male version of gametogenesis. This occurs in males seminiferous FEROUS tubules testes. Now there is a term which I will be talking about: spermatocytogenesis. Spermatocytogenesis. Now what is this? This involves stem cells. When the embryo is formed, primordial stem cells are there. Primordial germ cells, they are known as. They migrate and come to the pelvic region, and then there they either form ovaries or testes, depending upon hormonal triggers and other triggers. Sexual sex determination, if you have read, because of the presence of testes, determination factor, and all these things. So you have stem cells dividing to replace themselves because they are keeping on forming sperms. Stem cells dividing to replace themselves and to produce, this will produce a population of cells destined to become mature sperms. So these stem cells which are involved, they are the Spermatogonia, which we have talked about before. They are the Spermatogonia. So actually there are three functionally separate spermatogonial cells as seen. Three functionally separate gonial cells are seen. And they are classified on the basis of their appearance of the nuclei. The nuclei look different. You have the first is the type A dark spermatogonia. Type A dark spermatogonia. Then you have the type A pale spermatogonia. So this is known as the AD type. This is the type AD and type AP type. And then you have the third one. It is known as the type B Spermatogonia. Now, what does the type A Spermatogonia do? So, type A, basically, there's a population, many of them are there. So, type A actually, it maintains the population of Spermatogonia. If a amount of Spermatogonia decreases, then they directly they don't produce sperms. They don't get converted to produce sperms. They become something like type B, and then type B goes on to produce sperms. So AD1. So basically, they maintain the population of stem cells which will produce Spermatogonia. So let us draw the diagram. First, let us use this green color. Let this be the type AD Spermatogonium. Right? So what they do, they maintain more amount of this type A Spermatogonia. They multiply. They will give rise to more type AD Spermatogonium. They will give rise to the this is the type AP Spermatogonium. Pale one. And now this type AP Spermatogonium, they will give rise to the type B Spermatogonium. They will give rise to the primary spermatocyte. They will give rise to the primary spermatocytes. And the primary spermatocytes will give rise to the secondary spermatocytes. So let's see it in a complete scenario. You have type a population of type A. Some of these type A will give rise to type AP. The type AD will give rise to type AD. But many of them will regenerate the cells which are lost by getting converted into type AP. They will multiply and one population will keep on, we will replenish the population. The AD population. Then type AP will give rise to the type B spermatogonium. This will give rise to the primary spermatocyte. This will give rise to the secondary spermatocytes. Again, just revise the process of spermatogenesis. You have the spermatogonium, then primary spermatocytes. This actually you can call them to be the type B, right? It will give rise to the primary spermatocytes. That will give rise to the secondary spermatocytes. That will give rise to the spermatids. That will give rise to the mature sperms. Clear so far? Yes, no?
Yes, sir.
Yes, sir.
Okay.
Yes, sir.
Okay, let me push this up. We go to the next part. Then the next term which we'll be talking about, it is Spermatidogenesis. Now, what is this Spermatidogenesis? It is the formation of or creation of spermatids from spermatocytes. From spermatocytes during spermatogenesis. So, which step? Let's go back. So, this you can call as spermatidogenesis. Genesis. The next term which I will be using, it is Permeogenesis. Now, what is this Permeogenesis? It is the final stage of Spermatogenesis. Stage of Spermatogenesis. This will see the maturation of spermatids into mature motile spermatozoa. Maturation of spermatids to mature motile spermatozoa. So, if we go back, this step, spermatid, this is known as Permeogenesis. This is Permeogenesis. So far, are we clear? Yes, no?
Yes, sir.
Yes, sir.
Okay.
Yes, sir. Yes, sir.
Next, we got thing is that we have to see the structure of a sperm. If you see the structure of a sperm, on the top there is a structure which has got lots of enzyme. This is known as acrosome. It's known as acrosome. Then the sperm takes a shape something like this, a capsular shape. This is actually the sperm head. And below this is the nucleus. This is the nucleus. Then here you have a part region known as the proximal centriole. And now if you go to see the structure of it, you have the head piece, then the middle piece starts. And to this middle piece, you have the next centriole, which this long, this actually it has to be straight because I'm drawing very badly. This is the distal centriole. The mitochondria is very strange. You have a spiral mitochondria. The mitochondria is spiral. This is the mitochondria. Mitochondria gives energy. The spiral mitochondria. Then ending over here, there is another centriole over here. It is known as the ring centriole. This filament over here, this is the distal centriole. I'll just erase this part over here. I've drawn it wrong. This is the distal, and this filament which you see over here, this is an axial filament. It goes below also. This is known as the Men. And now the tail piece starts. This is the tail region. This is the mid region. And this is the head. And this is on top of the head is the famous acrosome. It contains enzymes which dissolve the membrane of the egg. So we'll have a look at the sperm structure as a whole. So I have not the tail goes on like this. You people have heard a lot about sperm tail. So it has got a tail. This mitochondria gives lot of energy for the tail to swim. Swim towards the egg. So are you clear about the structure of the sperm? Yes, no?
Yes, sir.
Okay. Now let us go. One question comes. Okay. A question comes. What are the changes? I think I have to increase the size. What are the changes accompanying the transformation of spermatid to spermatozoon or mature sperm? Or another way they can ask you this question: what are the phases of spermiogenesis? Phases of Spermiogenesis. So I'll just list down the names and then I'll explain. First is actually the formation of the head. Here you will find some changes take place in the nucleus. It takes structure of interphase nucleus. During telophase, you have the chromatid structure. It will take the structure of interphase nucleus. Right? It will have finely dispersed chromatin granules and a nuclear membrane. Finely dispersed chromatin granules, and it will also have a nuclear membrane. Then this is actually in the spermatid. The sperm has not yet formed. Then the nucleus will lose some water. It lose water and it will become closely packed in a small volume. I will draw these figures a bit later, but first let me explain what are the steps. In the nucleus, there is lots of RNA. It is removed and only DNA is there. RNA removed and only DNA present. So the water goes away totally. The next thing which happens after the formation of the head is changes in the Golgi. This is very important. Question comes. So we know that the acrosome of sperm derived from Golgi body. It is derived from Golgi bodies. So in the first step, early spermatid you actually have the membranes arranged concentrically around a small vacuole. Golgi membranes, G membranes, arranged concentrically around a vacuole. That is, if this is a vacuole, the membranes are arranged concentrically. This is concentric. Concentric means if something, this is a concentric circle. Arranged concentric, घुमा फिरा के. Then so you have lots of these vacuoles membranes. One of this, they start becoming larger. One of this. And what happens? And inside this vacuole, a small dense body known as the proacrosomal granule is formed. formed. Now let us start drawing a picture of this thing. I'm using a yellow color pen for this. So, first stage, right? Yellow color नहीं लगा. A stage. Let's say this is the nucleus. Right? You have this. I've drawn it very poorly. Golgi body. I remember a particular cartoon. Inside a cell, this is a nucleus. Then there is a structure which was drawn like this is supposed to be the Golgi body. A student had labeled this as Wi-Fi. So I remember this cartoon. So you have lots of small vacuoles here. Vacuoles or membranes. Membrane bound. Organized. Around a central vacuole. This is organized around a central vacuole. Now, gradually, if we go to stage B, one of this will start growing larger and an acrosomal granule will form. This is the nucleus. And one of them is growing larger. So, this is the granule. Then gradually the stage C, the nucleus and the acrosomal granule is formed. These are all the Golgi complex. Then we go to the next step. This is the nucleus. This particular granule will grow in size, and this Golgi, this will start reducing. The Golgi membranes will start reducing. This is known as the acrosomal vesicle. Gradually, the nucleus will become like this, and on top of this, the acrosome is forming now. And this is the this is reducing. The Golgi membranes are reducing. This is the acrosomal vesicle. In the next step, finally, the acrosome will be formed over here. Pointed acrosome. And the head will be formed. And this is the nucleus. You have the acrosome being formed. From the acrosomal granules, it started proacrosomal granules to acrosomal granules, and then it will form the acrosome. And this is the nucleus. Now let us look at what happens. The next part, what happens, next change which happens. The head has formed. Now you have changes in tail or changes in centriole and formation of tail. So let's draw the cell, the spermatid. This is the nucleus. Then you have the Wi-Fi or Golgi body over here. You have something known as an acrosomal sac. And you have lots of mitochondria. Mitochondria. This is the nucleus. And you know this is the Golgi. This will gradually take this shape. The acrosome has formed over here. Acrosome, this is the nucleus. Axial filament. You have the mitochondria coming over here. And finally, the mitochondria, the axial filament will go from the nucleus downwards. And finally, in the tail piece, you will find these mitochondria. They will organize themselves into spiral type. This is the formation of sperm. As you know, that this process, it is under the control of the hormone testosterone. So far everything clear today? Yes, no?
Yes, sir.
Yes, sir.
Yes, sir.
Okay. Then I stop sharing over here and I stop my class. I will send you this photo. Do you have any questions regarding this?
স্যার এই স্পাইরালটা টেস্টোস্টেরনের জন্য হচ্ছে তো?
না পুরোটাই টেস্টোস্টেরনের জন্য হচ্ছে। পুরো স্পারম্যাটোজেনেসিসটাই টেস্টোস্টেরনের জন্য হচ্ছে।
ওকে স্যার।
ঠিক আছে, তো দিস ইজ আন্ডার দ্য কন্ট্রোল অফ টেস্টোস্টেরন, দিস প্রসেস অফ স্পারম্যাটোজেনেসিস। টেস্টোস্টেরন না থাকলে স্পারম্যাটোজেনেসিস হবে না। নো টেস্টোস্টেরন, নো স্পারম্যাটোজেনেসিস।
ওকে স্যার।
ওকে,
ইয়েস স্যার।
okay, then I will maybe on we will try it on Sunday. Maybe we can have a class on Sunday, offline, online, and then on Thursday, again on Thursday we'll have offline classes in Asansol. ঠিক আছে,
ওকে স্যার।
Sunday
Yes, sir.
problem, Sunday say at 11 o'clock in the morning, 11 12 something like that. Oh,
না
Sunday
স্যার, সকালবেলা।
না, সকালবেলা সমস্যা আছে আমি জানি, নাফিসার ক্লাস থাকে পঙ্কজ স্যারের কাছে।
Yes, sir.
হ্যাঁ হ্যাঁ হ্যাঁ, তাহলে সানডেতে 6টার সময়, ঠিক আছে?
Okay, sir.
ওকে স্যার।
ওকে ওকে, ঠিক আছে, তাহলে আজকে ক্লাস বন্ধ করি। ঠিক আছে, ওকে।